Hey, welcome to a video for the second exam review for Mark Nielsen's Anatomy at the U of U. This video is specifically going to focus on digestive system. We're going to do a trace of food, and then we're going to look at histology, and we're going to look at some other questions that he may ask. So here's a sagittal head. You can see the oral cavity, oropharynx, the whole pharynx, the larynx, esophagus, nasal cavity, etc. Since we're doing a digestive trace, we're going to go through the mouth. We don't have an NG tube. We don't eat through our nose, so we're going to eat the non-weird way, just go in our mouth. There's the food. Food needs to go in our mouth. There it goes into our mouth, and we call our mouth the oral cavity. That's the first place for a digestive trace. So our goal here is to turn food into poop. We're not going to worry about absorbing nutrients. We'll do that later. This is just moving the food through our body and then excreting it out. So oral cavity, we swallow that back to the oropharynx. And then from the oropharynx, we move to the laryngopharynx. Now we have a choice. Do we choke ourselves or do we feed ourselves? Most of the time we're going to choose feed ourselves, so we're going to go through the esophagus. And we're going to switch to this picture. Esophagus is dead middle in the superior aspect of this image. I'm going to switch, this is Mark's picture. I'm going to switch to a more realistic. The esophagus is up in the top there. There's our donut in the esophagus. So from the esophagus we move into the fundus of the stomach, to the body of the stomach, and then to the pylorus of the stomach. Food doesn't just slip through the stomach like that quickly, but it gets churned in there for a few hours or however long it takes. So we include all three parts in this trace. After it's done and it's turned into chyme, we have this bolus that's going to move through the pyloric sphincter, which takes us into the duodenum, which is a C-shaped organ. So that donut looks like it's in the transverse colon, but you just have to imagine it's behind that. We just can't see it. So from the duodenum we move into the jejunum, which goes to the ileum, and then the ileum goes into the ceacum, so the first part of the large intestine. Now we ascend in the ascending colon. We move across the body with the transverse colon. We move down the body with the descending colon. And then we hit this shape, this S-shaped part of the colon called the sigmoid. And then before we exit, we hit the rectum. And at the very end, we have the anus. So there's our food trace from the mouth. So from the mouth hole to the anus hole. There's our digestive trace. Alright, so here we have a chart of the three-layered tube for the digestive system. This will be the basic outline for all the tubes of the GI. So we're going to fill this out, and then we're going to go through and see where we have modifications. Because Mark could ask you, here's the basic structure, tell me what the modifications are in this area, and then in this area, and then in this area. So first, let's just get this part down here. Alright, so we have the three tunics from internal to external. So anytime we are dealing with the inside of a tube that can communicate with the external environment, we're going to call it the tunica mucosa. And I'm just going to write T mucosa like that. So there's our tunica mucosa. In the gut tube, you can see there's 4 subdivisions, and we're going to move again from internal to external. So the lining of a tube is always some type of epithelium. So the subdivision here is just called the epithelial subdivision. So just put epithelium because it's the lining. And we know that the tissue type for this epithelium is just going to be simple columnar epithelium. The way I remembered that, simple columnar kind of looks like a bunch of French fries. Oops. Bunch of French fr I don't know. A bunch of French fries like this. And we're talking about the digestive system. So that's how I remembered the type of epithelium in the GI. Alright, so that's our epithelium. And then right under that epithelium, which we know is avascular, we're going to stick this layer called the lamina propria. So that's the subdivision. So lamina and then propria. This is just a thin little layer right under that epithelium, and it's going to supply blood to the epithelium. And we know our more vascular tissue is going to be loose connective tissue. So that's our tissue type here. Okay. And now the tunica mucosa actually has its own little muscular layer. This is to provide secretion or assist with secretion of stuff like in the glands of the epithelial lining. So we call this the muscularis mucosa. So it's just the mucous muscle basically. So muscularis mucosa or mucosae. It's a muscle, and we do not think about controlling this, so we know it's going to be smooth muscle. And then finally we hit the final subdivision of this tunica mucosa, and we call it the tila submucosa. This is just kind of a intermediate area between the tunica mucosa and the next area, and this is just going to be another loose connective tissue condensation. So we just call it loose connective tissue as the tissue. And that was kind of redundant to do that up there, but just ignore that. All right, so there's our tunica mucosa. We got the epithelium, lamina propria, muscularis mucosa, and tila submucosa. All right, so under that mucus layer, the tunica mucosa, we're going to throw the next tunic, which is the tunica muscularis. All right, so that tunica muscularis is it's going to be made out of smooth muscle, and that's going to be for both of these subdivisions. And we know that smooth muscle is avascular, so there's probably going to be some connective tissue with both of those smooth muscles. All right, so the subdivisions. The tunica muscularis has two muscular subdivisions, and they're called the circular layer and the longitudinal layer. And that's referring to the direction that the muscle fibers are going. So the circular layer is just going to kind of create this circle, kind of like layers of circles like this, where the longitudinal layer, its fibers are going to be more long, and in a cross section it's going to look like that. But if we flip that, those tubes are going to be long like this, and then the circular layer would look more like kind of like this. And just pretend that's 3D, but we're making like a tube here. So we have circular inside and then longitudinal on the outer surface. And then our final tunic, we're going to call it the tunica serosa. It's called tunica serosa because most of it is enveloped in one of those coeloms, the peritoneal coelom. The visceral peritoneum is the tunica serosa, and the coelom is a serous membrane, so they just named this covering the tunica serosa. This is just the outer covering of the organ, and we know that the serous membranes are always made out of mesothelium. Okay, so there is our basic plan. Now we need to know how do we modify this as we move through the gut tube. So let's just start at the esophagus. How is the esophagus modified here? So I'm going to write the tunica mucosa, tunica muscularis, and then the tunica serosa. Alright, so we know that we can touch our finger to the esophagus if we try hard enough. Also, there's a lot of hard things that can go down our esophagus, like if we're swallowing. So the esophagus is going to be lined with stratified squamous epithelium, and that's just for protective factors. So the tunica mucosa of the esophagus is altered. Instead of having the simple columnar, we're going to have stratified squamous epithelium. The tunica muscularis of the esophagus, the upper half of it, so the upper half, we actually have conscious control over that. So if we have conscious control over this muscular tissue, or this muscular tube, that has to be skeletal muscle. So the upper half is skeletal muscle, while the lower half is just that smooth muscle still. So that's how we alter that. And then we get to the tunica serosa, and this is not in that peritoneal cavity. Most of it, except for the very end tip under the diaphragm, is up in the thorax. So instead of tunica serosa, we call it this, tunica adventitia. And we've heard that term before, and we know if we see the word adventitia, it's going to be referring to dense, irregular, connective tissue. So the esophagus is kind of just a completely different tube tissue-wise, but it still has those 3 layers. So that's what we need to know about the esophagus modifications. So now let's talk about the stomach, because that's kind of our next part. Alright, so the stomach, and we're going to do the same thing. We're going to have the tunica mucosa, tunica muscularis, and the tunica serosa. Okay, so we're looking at how the tissues alter. So there's our esophageal tissues altering. Now we get into the stomach. The stomach doesn't alter too much from this basic plan. The tunica mucosa is simple columnar, and then we have all those sublayers. The tunica muscularis has an extra subdivision to it. So instead of only having circular and longitudinal, we also are going to have an oblique. We have oblique fibers. So those are just thrown in there with the circular and longitudinal, and it is still smooth muscle. There's no more conscious control of the GI after that upper half of the esophagus. So the tunica muscularis just has some oblique fibers in it. And then when we get to the more terminal end of the stomach, the circular muscle alters, and it creates this thing called the pyloric sphincter. That is still smooth muscle, and it's a modification of that tunica muscularis, and it's a sphincter, so that means we have this round muscle that can close off and get smaller. So that has to be a modification of the circular layer in that tunica muscularis. The tunica serosa, that stays the basic plan there. Let's go back to the tunica mucosa of the stomach. So there's no real tissue changes, but there are some structural features in here that are going to assist with increasing surface area. So the tunica mucosa has this structure called gastric rugae, and it's basically, here is the stomach. We have these little folds in here that allows the stomach to expand out if it needs to. If we eat a bunch or there's some gas building up, it allows the stomach to kind of expand, and that's our gastric rugae. Alright, so moving from the stomach, and we go through that pyloric sphincter if we're doing a trace, the next part we're just going to call the small intestine, because these three organs, the duodenum, jejunum, and ileum, they're all going to have the same kind of basic layout. So I'm just going to say small intestine. And you may also hear this interchange with bowel. So small bowel, small intestine, same thing. Let's look at what happens here. So we have the tunica mucosa, tunica muscularis, and the tunica serosa. Okay. Tunica mucosa, the tissue stays the same, but we're going to throw in a couple structural modifications again. So these are not tissues, these are structural modifications. So the tunica mucosa in the small intestine has these structures called circular folds. The function of these is they increase the surface area. And we want that to absorb as much nutrients as we can. So we get those circular folds, which increases the surface area a ton. Lots of nutrient exchange here and absorption. So we have circular folds in the tunica mucosa. And then we also have these structures called intestinal villi, which same thing, they increase the surface area of that small intestine, because that's where the majority of nutrients are going to be absorbed into the body. The tunica muscularis, exact same as the basic pattern, so no real changes here. And the tunica serosa, also same thing, no real changes here. So the big change is in that tunica mucosa layer. Then we're going to move into the large intestine. You may also hear this called the large bowel, same thing. So our large intestine, we have that tunica mucosa, tunica muscularis, and then tunica serosa. So as you're studying, just go ahead and make a chart like this, like do a digestive trace and follow the food, and next to that trace, just put this is what's being modified here, etc. So you just want to get used to knowing what's modifying. It's really not too bad. Alright, so let's look at what structural modifications here. There's no real tissue changes anymore. The tissue changes are really between the esophagus and that stomach. So large intestine, structural modifications. Instead of circular folds, we're going to have these folds called semi-lunar. So instead of a full circle, we're going to have these kind of crescent-shaped semi-lunar folds. There's not as much nutrient absorption in the large intestine. This is more where we're going to suck in water from the gut or kind of balance our fluids in the body here. Insert or exit water. So we have the semilunar folds in the tunica mucosa. The tunica muscularis, we have a couple modifications here. The longitudinal muscle of that tunica muscularis creates this structure called the taenia coli. Taenia coli. The word taenia just means like a band. So we have this band or ribbon on the colon, and that goes across the whole entire large intestine. And that just helps with peristalsis, moving whatever's inside of the large intestine through the tube to eventually exit the body. And there's these little pouches called haustra. These are kind of remnants of the peristalsis. So there's these little pouches of the large intestine. That's what haustra is, just little pouches, and it's in that tunica muscularis due to the muscle contractions. And then the tunica serosa, where we see the Taenia coli, it actually pinches out a little bit of fatty tissue. So we have these things called fatty appendages, and you may also hear this called appendices, but either word works. So fatty appendages in that tunica serosa, just little fat stores on the outside of the large intestine. So there's our structural modifications. So let's just kind of do a recap here. So our esophagus has a lot of tissue modifications. So we see that tunica mucosa, stratified squamous instead of the simple columnar. The tunica muscularis has skeletal muscle, and it's not just full, smooth muscle like the rest of the gut tube. And we don't have a tunica serosa, we have a tunica adventitia, made of the dense irregular connective tissue. That stomach, the tunica mucosa has a structural modification. So let's say if I highlight it in red, it's a structural modification. That means the tissue's not changing, it's just a structural modification. So the tunica mucosa has gastric rugae, which is a structural modification. The tunica muscularis has the modifications of having oblique fibers and also that pyloric sphincter. So there's our structural modifications of the stomach. We move to the small intestine. Here's our structural modifications, the circular folds and the intestinal villi. Both of those just act to increase that surface area. And then the large intestine, structurally, we have the semilunar folds. It's along the whole entire large intestine. The muscularis has the Taenia coli, and then the little haustra pouches, and then the serosa has the fatty appendages. So there's structural modifications. There's not a lot of tissue modifications throughout that whole GI. So another question Mark may ask us, after we know these modifications, he could ask us, what are 4 structural modifications that increase the surface area of the gut tube? Whoops, gut tube. So now we should know that. So does the esophagus have any? No. Does the stomach have any that increase surface area? Yes. That was called the gastric rugae, or gastric folds. Gastric rugae is one of them. Does the small intestine? It does. It has the circular folds as well as the intestinal villi. So intestinal villi, circular folds, increase the surface area in the small. Then we get into the large, where we have these semi-lunar folds, still increasing surface area for the water reabsorption, or absorption, sorry. So there's our 4 structural modifications that all increase surface area. Alright, so here we have this picture that's commonly seen on exams. He'll give you a kind of coloring key like this and have you color all these different structures. So let's just make sure we can all do this so we can get those points. So first thing we're going to color is the epithelium of the tunica mucosa. So I'll go ahead and do that. Okay, so before I finish up here, this last area over here, there's kind of this circle over here that you need to be careful with. So you can see there's a line in the middle of this circle. I'll show you what we need to do here. So zoom into this. So half of this is going to be one thing, half of this is going to be the epithelium. So just really carefully, this upper portion here, that's part of the epithelial. It's like a weird cut. You're seeing the inside somehow. But just know you want to color that top part of that circle pink if you're using pink, which I would recommend. Okay, so we've colored all of our epithelium now. We're going to the next layer, and we call that the lamina propria. So here's our lamina propria. Definitely get a bigger slice here. Okay, so the lamina propria is always deep to the epithelium, and it's where the blood vessels are going to run in the villi. So what we're going to draw or color this as is right here. We can see we have some blood vessels and lymphatic vessels here, and these are just, this color is just saying this is loose connective tissue surrounding those, bringing the blood vessels. Obviously, you don't want to color in the lines. Okay, so as we're finishing this up here, see I'm right under that epithelium. And we can see there's these little holes in the epithelium. Not totally sure what those are supposed to represent, but they are going to be colored green. They may just be areas that the villi have been cut off. And then we zoom in here, around the blood vessels you color green, and the lymphatic vessel. And then here, this tiny bottom part of that sliver, that's also the lamina propria there. Okay, so now we have the two layers colored here. Next layer we're going to go to is our muscularis mucosa. So this one is pretty straightforward. I'm going to do it in this darker reddish, light red color. It's always under the lamina propria, so I'm just going to I'll color that in here. Alright, there's our muscularis mucosa. Then finally we get the tila submucosa. I'm going to use a light blue color for that. Alright, so the tila submucosa is another loose connective tissue. We know that's going to be where our blood vessels are found. So in this kind of blank hollow space under the muscularis mucosa, this is all the tila submucosa. So you kind of want to go around all the blood vessels, and then it's kind of just this box in this area. Color on this black, darker surface. Alright, there's the tila submucosa. So we can see we've worked our way through those 4 layers of the tunica mucosa. Now we are looking at the lymphatic capillary. So the lymphatic capillary, I'm going to do in this orange color. The lymphatic capillary is this one that projects up into the villi. So that is our lymphatic capillary. And that's the one that projects up into the little nipple structure. That's also called a lacteal. So you might hear that interchange, lymphatic capillary or lacteal. But that's going to kind of suck fats out of the GI tract and then return it to circulation. So there's our lymphatic capillary. Then we get tunica serosa, which we know is the outermost part, outermost covering. Okay. back to the tunica serosa. So the tunica serosa, we're going to find the outside of our tube, turn this a little bit thinner, and there is our simple squamous epithelium, or our mesothelial cells out on that surface. Again, you'll want to try and stay in the lines when you do this. Okay, now we have our muscle layers. I'm going to do the circular muscle in this red color. Make the size bigger here. So red. We can remember the circular muscle layer is the deep layer. And in this, the cells look longer, but that's because we're looking in this tube, and the cells are going to look longer in this view because it's moving in the circular direction. So here's our circular muscle. Alright, there's our circular muscle. Next, we're going to have the longitudinal muscle. I'm going to do that in purple. So here they look like little eyeballs. That's because they're moving lengthwise in and out of the screen. Alright, so now we've pretty much colored all of our layers. There's a couple things that I myself forgot to do. So I'm going to go back to that lacteal, and right here in the center, that is also a lacteal that I did not color. So always make sure you colored everything. I don't know if that's the same color. So just in case, I'll put it, yeah, it's not. That's okay. So there's our lacteals. Then we have a capillary. So the capillary is the vessel that projects up into the villi. So I'm going to use this neon green for the capillary. Okay, and then also we're going to come over to this little one. Those are both our capillary here. So we want to make sure we color those little guys also. And then finally, let's see, lymph vessel. So the lymph vessel is where that lacteal is coming off of. So I'm going to draw this color like this purplish. Again, maybe. All right. And then finally we get our blood vessels, and I'm just going to do that in whatever color I can find that I have not used. All right, so there you can see I've colored everything. And there's what you want it to look like. And yeah, hopefully that's not on the test because it's a lot of coloring, but it shouldn't be too hard if you have practiced a couple times. Shouldn't be too bad. All right, so to end off this super awesome lecture on the GI, we're going to talk about those 3 organs that assist with digestion. So we're looking at the liver, pancreas, and the gallbladder. We want to know what these do. So this is what you need to know. The liver produces this stuff called bile, which just kind of is a basic substance because the stuff coming from the stomach can be acidic. So we throw in some bile to give it a little more alkaline. Just know it produces bile. You don't need to worry about the other stuff I just said. Produces bile. The pancreas produces digestive enzymes. It also produces this stuff called insulin. That could be another thing if you want to include that. And then the gallbladder is going to store bile. So the liver produces bile, the gallbladder is going to store that bile. So let's kind of look at what this is, how these are all connected. So here is a diagram of the liver. Liver's in the upper upper right quadrant of the body, and then we're going to have this other little organ hanging out next to it right here. That's what we call the gallbladder. And in real life, it's green. You open a body, a lot of the time, the whole body on this right area is soaked green, just because the gallbladder can sometimes pop. And then we're going to have the pancreas kind of down here. So there's those three organs. So somehow they have to put their stuff into the gut. So I'm going to draw, I'll use blue here. We're going to have the duodenum, which is this kind of C shape as it leaves the stomach. So that blue is our duodenum or duodenum, and that's coming from the stomach. There's our pyloric sphincter there. All right, so all three of these things are going to connect into one common duct system to dump that bile and digestive enzymes into the duodenum so we can break down that food. So coming from the gallbladder, we have this duct like this, and that's called the cystic bile duct. So that's the cystic bile duct. Cystic bile duct. That's coming from the gallbladder. Then we're going to have this other duct that's coming from just the liver. We call this the hepatic duct, and they kind of meet right there. But that's the hepatic duct. And then after they combine and they create this other duct, this light blue, that's called the common bile duct. So that blue is the common bile duct. All right, so that's how we connect the gallbladder, the liver, and now we're going to draw the pancreatic duct. So pancreatic duct kind of starts over here, and then they all join together there. And then they're going to dump into the duodenum like that. So that's the main feature here, is that these three organs start separate, but then they all dump into one common thing, that pancreatic duct. And then they dump into the duodenum to help break down the food. Alright, so now that we've looked over those organs, let's do a trace. And this is something I haven't seen Mark do, because usually these exams are not together. But let's just look at what this could look like potentially. So he could say, you have food, you swallow it, and then you're going to absorb glucose in the jejunum and take that glucose to the heart. Okay, you should know how to do this. It's not that hard now. So let's just swap. Let's just start that. So here's our just normal trace. From the esophagus, we move to the stomach. And I'm going to include the three parts here. So we're going to have the fundus, the body, and the pylorus. Okay, after pylorus, we move that through that pyloric sphincter into the duodenum, into the jejunum, which, if you remember, that's where we were going to absorb that glucose. All right, so here's where the absorption is going to take place, and we do that through a diffusional barrier. Again, diffusional barrier here. We have to know what lines the jejunum. So We're in the digestive system, so we're going to say simple columnar epithelium. And since we're absorbing something, we're going to move into the cardiovascular, which I know is always going to be endothelium. All right, so now we are moving through that endothelium and we're into a capillary. He doesn't teach us the names of these capillaries, so I would just put capillary of jejunum, and then we'd have a venule. But then we do know what the vein is going to be called. So the jejunum is part of the midgut. The midgut is drained by the vein called the superior mesenteric vein. Superior mesenteric vein. That superior mesenteric vein goes up into the hepatic portal vein, which goes to the hepatic sinusoids, which exits the liver via the hepatic vein, and then to the inferior vena cava, and then we would be at the heart. Cool. So that would be absorbing glucose. You swallow something and you absorb it and it gets to the heart. He could definitely do something like that.